Welcome back. The topic for this video now, we're still on the topic of gene transfer, but now we're going to go over what the 4 types of gene transfer are. And in this video, I'll talk about the first one here on the list, conjugation. Okay, so this is going to be one method by which bacteria can transfer bits of DNA to other bacteria to essentially increase the genome or the DNA that's present within that bacteria. Okay? So we've got conjugation. We've got transduction, which is going to involve the use of a bacterial virus in order to transfer DNA. We will talk about transformation, which is going to be kind of neat because this is the type of bacterial gene transfer that we as scientists exploit in order to make different proteins that are needed. Okay? So we've got conjugation, transduction, transformation. And then the last one I'm going to put in a different color. And I'm going to put it in a different color not because it's more or less important. Oh, this is a particularly squeaky marker. But the last type of gene transfer are transposable elements, okay? And those transposable elements are often referred to as gene jumping. And I've got it in a different color because while there is some potential evidence that transposable elements might be present in bacteria, they were first found in plants. And so they're more associated with eukaryotic rather than prokaryotic cells. But it's still really cool how this happens genetically, so I'm going to keep it in here. Conjugation, transduction, and transformation are going to be the 3 modes that are going to be most common for prokaryotic cells, though. Okay? So keep that in mind. We'll talk about first conjugation. And with the process of bacterial conjugation, there is going to be an F-positive cell. This is going to be the cell that has the plasmid to be transferred. We've got an F-negative cell. That's going to be the one that lacks the plasmid. All right? Primarily, conjugation happens in gram-negative bacteria, but it can happen in others. But usually, it's associated with gram-negative bacteria. So I'm going to draw some rod-shaped bacteria here. And the pink one, I apparently need a new pink marker too, is going to be our F-positive cell. And the green one is going to be our F-negative cell. All right? So each of these are going to have genetic material that makes that particular bacteria what it is. All right? So it's going to have its genome. But the thing about our F-positive cell, this is going to have that little bit of circular DNA. This is going to be the plasmid. Okay? And that's what's going to be what is transferred to the F- cell. All right? So what we have with bacteria and the process of conjugation, I'm going to draw it over here just because how I drew the 2 bacteria. So what you're going to have with the conjugation is the formation of pili. Okay? Sometimes it's not pili, sometimes it's referred to as a mating bridge. You'll see either of those terms used. I think pili is what I introduced the structure in a previous video when we were talking about structures that are associated with some bacteria and not others. So the pili are going to essentially exist with the bacteria that is F positive, because the F positive bacteria is going to be the one that initiates this process. It's going to establish a connection with the F negative cell, and that connection is going to be the pili. So that pili is essentially an empty tube that connects the cytoplasm of the two bacteria. And then what happens, that plasmid is going to transfer across to our F negative cell. Once that plasmid crosses, our F negative cell now becomes an F positive cell. So the information for establishing that pili is contained within the plasmid itself. So that offers and confers the ability of next that bacteria to be able to pass on that little bit of DNA. All right. Along with that ability, that's where you get additional genes that are going to be transferred on that plasmid, because there's going to be more than one gene associated with the plasmid, and some of those genes that are associated with the plasmid are going to be things that are advantageous for the bacteria, either being able to grow in environments that it normally can't, conferring additional advantages onto the bacteria, like virulence factors can also pass via a conjugation bridge or mating bridge in the case of the pili. So what you've got with conjugation you get a direct transfer of the DNA from one bacteria to another. And that transfer is going to happen via the cell structure, the pili. So conjugation, this is the genetic process where pili are important, okay? So what happens with that, you get the crossing over of genetic material there. The thing with conjugation, and what you'll see with bacteria oftentimes, you will see a high frequency you'll see some strains that have a high frequency of recombination. So if a strain is designated HFR, it's indicating that it has that ability to generate the pili and pass on genetic material. Okay, so with that high frequency recombination, the genetic material and the transfer of genetic material is just going to be more likely. So when bacteria are classified as HFR strains, it means they have that ability to transfer information to other bacteria, and it happens at a fairly high rate, okay? But with the bacterial conjugation, the important structure there is going to be the pili, and that plasmid is going to transfer and move across that bridge, that conjugation bridge or that mating bridge, to transfer the genetic information to the cell that doesn't have it, okay? Hopefully that all makes sense. If you have any questions, let me know. The next video, I will talk about the process of transduction.